So, hello everyone. I'm Haejun Han, and I'm a PhD student in bioengineering department. And I'm really excited to introduce our speaker for today's breakfast seminar, Dr. Eva Dyer. She did her undergrad at the University of Miami and her PhD at Rice University, both in electrical and computer engineering. And as many of you already know, she's an assistant professor right here in the Quarter Department of Biomedical Engineering at Georgia Tech and Emory. Dr. Dyer and her research are both super cool. She's working on this awesome intersection of machine learning and neuroscience. She's trying to figure out how neural circuits and neural computations work and how we can use that knowledge to make AI systems better and more flexible. And her accomplishments have been recognized with a lot of awards, including the Sloan Fellowship, the Next Generation Leader Award from the Allen Institute, and CFAR Global Scholar in the Learning in Machines and Brain programs. And I have the chance to have her on my Quarks committee and thesis proposal committee, and hopefully in my actual thesis committee in the near future. And her lab and our lab have been collaborating years, and it has been always a pleasure to talk to her. She's not only very brilliant, but also very nice, kind, and fun. So today, Dr. Dyer is going to share her latest insight with us in a talk. Actually, I heard that the talk title changed from the flyer. So yeah, let's see what she's bringing here. So let's give a big round of applause and a warm welcome to Dr. Dyer.
Thank you so much, Haejun, for the really kind introduction. And thank you all for making it out for this early morning session and having breakfast. And I'm excited to be talking to folks here, at Georgia Tech about the work that we've been doing over the past few years. This is just kind of an intro slide, just talking about the sorts of things that we do in my lab kind of broadly. The lab is called the NERDS Lab, or Neural Data Science Lab, and that sort of encapsulates, the two pieces. So we're thinking about how do we develop and leverage the kind of growing explosion of AI and machine learning methods. How do we do this, and then apply that to neuroscience to tackle a number of different questions about neural computations. And then we're also, of course, thinking about how do we leverage insights from the brain and neural computation and bring that back to machine learning as well. So we have the arrow moving between AI and neuroscience. Within machine learning and AI, we do a lot of work in representation learning, so things that involve learning from data sets where we have maybe limited amounts of labels, maybe no labels in the case where we have a fully unsupervised problem. And really our goal is to be able to take these complex data sets and distill them down into representations that allow us to interpret or build insights or inferences about the data. And then with respect to neuroscience, we're really interested in, a number of kind of driving questions that we're investigating with our collaborators. A big focus has been on, how do we take these data sets, observations of the activity from neurons or different regions in the brain, and how might we be able to build biomarkers or understand things about when neurodegeneration or other disease starts to manifest in those neural circuits. So how does neurodegenerative disease impact neural dynamics? We're also interested in questions that have to do with comparisons or individual variability. So if we look at two individuals or two animals carrying out the same behavior or the same movement, how is that encoded by the brain, and how is it differentially encoded across different neural circuits? And then, questions about that we've been more recently investigating about exploration, exploitation, or different decision-making strategies in the brain as well. And we're really excited to kind of develop new tools to tackle a number of different neuroscientific questions. So that's kind of just like a big picture view. And then what I'll kind of focus on today is some work that we've been doing in this space of representation learning, AI for neuroscience, for neural activity and neural decoding. And specifically, what I'll talk about is, oftentimes we have recordings from populations of neurons in the brain, and we want to try to make sense of it, right? Like, what are these neurons doing? How are they communicating? How are they building certain percepts, sensation, action? And so over the past decade or so, there's been a lot of work, including a lot of folks here at Georgia Tech, in trying to understand how populations of neurons or neural circuits are actually working together. And so what I'll talk to you about is some ways that we've been kind of pushing on this idea where instead of kind of throwing away or instead of only caring about what the population is doing, we also have ways to reveal what individual neurons are doing as well and how they're contributing to different computations. So seeing the forest and the tree, being able to see those individual contributions in addition to the, the bigger picture of how everything comes together. All right. Cool. So with that, I'll just kind of dig deeper into some of the motivation behind our work. So, there's been a lot of really exciting opportunities, both due to growth in neural recording technologies, so our ability to measure, more neurons from, larger fields of view or across multiple brain areas. We now have, in terms of experimental methods, capabilities to label specific cell types, which give us a lot more information about some underlying functional properties of these neurons. And then in addition to kind of recording tools that allow us to see bigger and bigger fields of view or more and more neurons over more and more brain regions, there's also been a lot of innovations in behavioral tracking and analysis that allow us to now put animals and their brains in more kind of naturalistic environments and try to record the brain while it's, doing things that it would normally do in relevant settings. And so more complex behavior, and then in addition, we're also able to now record over very long periods of time from the brain. And so this has also given us a new lens into things like aging and learning, where you really do have to record chronically over long periods of time to understand what's going on. So with all of these new sources of information from the brain, we're really faced kind of in some ways with a bottleneck in our interpretation of those data sets. And we need new ways of modeling neural activity at this scale. So that's really been a big driver for the research that we're doing, is thinking about how can we really build robust ways of reading out from the brain, and how do we do this in such a way where we could scale to really large recordings across really diverse, different sets of neurons, potentially. Okay, so we have all these data sets, and so obviously there's a number of challenges that we're facing in trying to build these, new models of brain activity. And so there's a number of them that, some aren't unique, I guess, to neuroscience only. But I'll kind of talk to you a little bit about some of the things that, we think about and the challenges that we're facing within the field. So the first being just that, when we measure, so when we go in and we record from, neurons within the brain in a particular, say, brain region, even when we, say, present the same stimulus over and over, to the brain circuit or to that neuron, or when we see the same behavior basically carried out by the individual, we actually see that there's not a clear mapping between neural activity and behavior, right? So there is a lot of noise and variability in neural responses that make this problem in terms of a deterministic mapping quite challenging. So then in addition, right, we have the ability now, instead of just looking at a single neuron, to now look at, say, a population of neurons, as I sort of discussed. But then one of the challenges there is each time we go and record from the brain, even if we have, say, a chronic recording where we have an implant where we're kind of monitoring the same volume of tissue, we actually find that, you know, the number of neurons or the population of neurons that we might have access to can also be changing. And so for this reason, transfer, or the ability to kind of take recordings from, say, one day where we're measuring the activity from the brain, and then transfer that, or kind of take what we learned and map it on to a new set of recordings, is incredibly challenging. And that's kind of just shown in this picture here, where, you know, in, say, one session of a neural recording, we might have access to this subset of neurons, and, you know, that's the ordering or the set of measurements that we get. And that at another point in time, maybe we have access to this other subset of neurons. So essentially, in addition to having variability in the neural responses and their mapping to stimuli or movements, we also have this challenge where the recordings that we get are often a kind of not necessarily random, but a sparse or a different sampling of the underlying sources of information that we're trying to access. Okay, so in current sort of ways of interpreting and representing this data, what do we currently do? And so I think that the challenge of transfer hopefully will become more clear as I sort of show this picture of how we want to actually represent this population of neurons and the activity therein. So we can think about, essentially here, I'm displaying the activity of multiple neurons that are spiking action potentials, the main kind of source of information within the brain or amongst neurons. And so here I'm just displaying the activity of different neurons that are all firing simultaneously and at this population level over time. And so in order to now take this whole ensemble of recordings that we can see and we can measure, and then think about how they're interacting and maybe coordinating towards some sort of collective dynamics. The typical way that we can think about this is essentially if you have, say, D different neurons all firing at some underlying firing rate, you could think about slicing through at a given point in time and now representing the firing rate of all of these neurons as some point in a D-dimensional space. So this is sort of shown here in just a three-dimensional picture where we can think about the activity of neurons at a given point in time as being like a point on some underlying potentially low-dimensional manifold within this high-dimensional state space. And the idea is that because neurons are actually coordinating and kind of firing in these correlated patterns, this manifold or this kind of the state space that they occupy isn't everywhere, but it's actually kind of often constrained to simpler manifolds depending on what the underlying behavioral structure and variability looks like. So we can kind of think about then taking the activity of multiple neurons, they become a point in this state space, and now we're thinking about the evolution through this sort of low-dimensional manifold as being the evolution of these like neural activity patterns manifested here. Any questions just about, I know that this is a broader audience, so trying to make it accessible. Yeah, go ahead.
So that state space, is it just, for instance, if you have 3 neurons, is it just spiking rate of each neuron? So like like three-dimensional, or it also contains the information about connectivity of those neurons?
Great question. So in this picture, we're just going to consider the firing rate. There could be some other way of representing the spiking activity, but yeah, we'll kind of consider an estimate of how many spikes per bin, and then that will be the coordinate, one of the coordinates in this state space. So the first is you described.
Then is the connectivity of the neurons, that information is embedded in the shape of the space, maybe?
Yeah, it's a great question. So like this doesn't assume that we know anything about the connectivity, but of course neurons are interconnected, and that's what's going to potentially drive these correlated activity patterns, either through interactions or also maybe through driving inputs as well. And so there are, other sets of techniques that are used to maybe try to infer those connectivity patterns also from population activity. I actually will, so once we get further into the talk, you'll see some ideas and some tools that can start to give us a lens into some of that, but it's a really interesting and kind of outstanding question of how we put all this together. Thank you so much, yeah. Yes?
What's the data source? How are you reading neurons? Is it functional MRI, or are you actually?
Oh, yeah. So here I kind of didn't go into necessarily the details of some of the different ways that we can measure or record information from neurons. So in a lot of what I'll show, we'll consider multi-electrode probes, so different sorts of Utah arrays or linear probes that allow us to measure the electrical potential, or essentially see action potentials from neighboring spikes after spike sorting, which we won't talk about. But the data format will kind of look like this, where we're reading out, the activity from neurons in a kind of nearby region from the probe. But a lot of the tools and things that we'll talk about can also be applied to calcium imaging. So there are other ways of, imaging large populations of neurons. And some of the stuff that I'll talk about later will also apply to calcium imaging. So, yes?
In the data set, then each data point is a single neuron, or is it like a single area?
So in this, we'll assume that after pre-processing of the data, we've done a pretty good job of isolating individual neurons, or there could be cases where you might have more than one neuron that's sort of grouped together into one of these different channels that we're measuring. But yeah, the idea is that we've kind of sorted the data so that we've identified for the spikes that I'm recording on a probe, which neuron those spikes were associated with. Thank you for the clarifying questions, yes.
Can these models in principle be scaled to coarse-grained recordings like EG, LFP, and similar, or they are designed for single independent, let's say, units?
Yeah, no, so the perspective that I'm showing here of taking multivariate time series data and kind of treating it potentially, through this lens of looking at, what is the representation, or what is the kind of embedding at this point in time, and how can I, take multiple channels to form some sort of embedding. Like that can be applied to any multi-channel time series, so EG. But some of the challenges that I'll talk about specifically, in particular this idea of transfer, and the fact that each time you're getting a recording, you're actually seeing a different subset of the underlying, let's say you have a fixed population that is the circuit that you're trying to measure. This challenge that each time you're actually measuring from different channels, and the channels aren't necessarily aligned across time, that is I would say, unique problem that we kind of face within electrophysiology. Just because EEGs are a little more, standardized, right? You have like the same coordinates, and so you know that, in this recording versus the next, or an fMRI, right? You have a voxelized sort of head space where all of the points in space are kind of aligned to each other. So here we're facing this problem where across different days, across different sessions, we could have subsampling of the population as well as permutation of the units. So even if you have the same units that you're recording from, you don't necessarily know which input channel is the same from day to day. Okay, thanks so much. This is good because there's a lot of ways that we can measure activity from the brain, and yeah, some of the challenges are shared and some are unique. Okay, so we were at this picture. We were kind of thinking about the neural activity at a slice in time as kind of forming some picture in this state space. And then, part of the goal is to learn this underlying low-dimensional manifold and understand like trajectories and dynamics through this space. Okay, so this is just taking the same picture here and just showing it again. Here, just showing this idea where you have 4 neurons in this case. You're now taking their activity as this sort of 4D, representation in the state space, and then you're looking at how their collective activity is evolving from one time point to the other, right? And so that's what we just saw on the last slide, shown here. And then I'm not gonna have too many equations. This is, I think, one of the few. So we can basically think about this if we were to say, learn some sort of mapping from this neural space onto some behavior or some sort of target. In the stuff that we'll talk about, we'll think about decoding different movements from the motor cortex of the brain, for instance. Okay, so we have all of these different neurons, and let's say we have N neurons, right? And so we're finding an embedding at a point in time based upon all these N neurons, and then we're gonna learn that mapping, right? And this is just the normal setup and what I've described. And so now, what I said before about the ordering of neurons is really important. So if I want to learn a mapping F from a set of N neurons, and now I want to somehow, on a different day, apply that same embedding or that same F to a new set, if I have a different ordering of my inputs, then whatever I learned is actually not useful to me any longer unless I have to retrain or relearn like some rotation of my space to make these two things kind of useful. So the fact that we learn a mapping from the n-dimensional space down to behavior or the thing that we want to read out can be limiting based upon the arguments that we said before, right? Where we have a different set of inputs at a new session and we can't actually use our same F. And so in order to kind of tackle this problem, we recently developed a model that allows us to basically separate time and space in order to kind of resolve this dilemma in some ways. Okay, and so what does this mean? So now instead of learning one mapping across all of our inputs, we're instead going to just separate this function into two parts. The first being a mapping that just applies to individual neurons, right? So each time series that I pass in basically gets exactly the same mapping, right? So I'm gonna apply F onto just the time series of individual neurons instead of applying it jointly to all of the whole population at once. So now I'll say I'm gonna learn an F onto each of the different neurons. It's exactly the same mapping. It doesn't know which neuron's which. So you can apply this, independently. And now, to get to my final kind of population level description and encoding or output of the behavior, I then am going to learn a secondary function that does care about the order of these things, but it can now learn how to put together all of the individual pieces into a kind of global description of the dynamics. And so that picture can kind of be shown here, where we saw our picture before, where we had these 4 different neurons, right? And we're just going to group them together. We don't know which one is which. We don't care any. They're all, they don't have any identity in some case or in some sense. And then we have this blue neuron. Now we're going to model its dynamics, its independent dynamics. We don't know anything about the red and the green and the yellow, and we'll do that the same for each of our independent channels. And then in this second stage, we're then asking, okay, well, how do all these things kind of fit together at the population level? And we do that actually kind of independent of time. So when you learn these 2 mappings, you kind of learn how to separate the embeddings of these individual time series from the population level description. Okay, so that's the kind of main idea, and I'll describe how decomposing it in this way actually allows us to solve some important aspects of that transfer problem that we described before. Namely that now we've learned an encoder or an F that actually can operate independently of which neurons are passed in. And so potentially what we've learned in F can be transferred to any new set of neurons without any retraining. Okay, so how do we do this? So the model that I showed, this decomposition across time and neurons or time and space, could be implemented with a number of different architectures or building blocks. But in this case, we use transformers, which are actually a very flexible sort of building block for neural networks, for deep learning, that allow us to learn non-local interactions across different pieces within a sequence, or in our case, I'll show, we can learn non-local interactions in time, but we can also learn interactions in space, so across different neurons with transformers. How many of you have already heard about transformers or are somewhat familiar? Okay, so I'm sure a lot of you have been excited about the recent advances from ChatGPT and the sort of things that have been enabled by these really powerful architectures in deep learning. And so at the heart of a lot of those advances are these tools or building blocks for neural networks called transformers. And so really, we can think about, and a lot of this initially was sort of pioneered in the space of natural language processing. So there we can think about, a sequence, right, of text or I guess I won't have tons of time to go into all the all the background of transformers so that I can get through a lot of the content here. But I'm happy to talk more about this in detail with those of you that are interested. But so typically we think about a recurrent neural network or something that has temporal or the ability to process sequences as having a somewhat local view of the data, right? And so there is some encoding of the history that is fed into, say, an RNN or LSTM. But within recurrent neural networks, it's often really hard to form associations that are very non-local. So if there's something in the sequence that you care about that happened at the beginning, or I guess if it's causal, you don't know what happens at the end. But like kind of existing sequential models couldn't handle that ability to form those more non-local interactions. And so what transformers allow us to do is through a mechanism called self-attention, essentially for each point in your sequence, you can figure out if there are other places in the sequence that are important to you for making your decision. And so sort of visualized here, attention is all we need. And so you see as you move further in the sequence, right, if you're at the end of the sequence, the other words that you care about are pointing more towards the beginning. And so these weights are sort of visualizing for each point in the sequence what other things you care about and what other things are you attending to in the rest. Okay, so that's just at a high level. Transformers for sequences are really the kind of backbone of a lot of the current, exciting innovations within deep learning. So, okay, so we'll use this as basically, I'm going to skip ahead. We'll use this as a building block for our separable transformer or our separable architecture that I sort of described. And how do we do that? To begin, we're going to first focus on this, the F, right? The first function that's going to process our individual neurons and their time series. So that function is trying to find some embedding of each neuron based upon how it's firing and what its pattern of activity looks like. And so we'll basically, instead of each word being a token in our transformer, instead of attention is all you need, we'll take the time series for our neuron and we're going to kind of bin it. So now our tokens that we're learning our self-attention over are just the firing rate of an individual neuron over a small chunk in time. And so we're going to feed that time series for each neuron into a transformer. It should be able to learn some patterns, from those sequences that allow it to take each neuron and find some representation or embedding of it. Okay, and so now each neuron has a representation, or it has some sort of information extracted from it. And then in this second stage, we're now going to also apply self-attention, but now it's not a sequence, it's across different neurons. So we don't necessarily know which neurons are close to each other. And there are ways to deal with that within transformers through learned positional embedding. So instead of having, placing here, we know exactly that this time point comes before this one, and so on and so forth. In the case of our interaction across neurons, we don't necessarily know that. So that's something that we can actually learn through this sort of self-attention model, but now instead of thinking about things happening over time, you're saying, which other neurons do I need to pay attention to in order to maximally understand how to decode some movement or solve my underlying decoding task? Does that? Cool. So so we basically have two transformers, and we can kind of combine them together, where we'll learn in time and learn in space. Okay. And so what's kind of cool about this, going back to our earlier challenges about being able to transfer across different days, or maybe even transfer across different animals, so different brains, right? We have this ability through this decomposition to learn on one set of neurons and actually freeze our model and see how well the first half of the transformer applies across a new set of neurons. And so we could potentially do this across brains as well. And that's what I'll show next. Okay, so we applied this model, we've benchmarked it, compared against a number of other kind of competitive models. So the NDT is actually from Chetan Pandarinath's lab here at Georgia Tech Emory as well. So the neural data transformer is sort of taking the original state space view that I talked about, where we're going to essentially represent each point in time as a sort of vector of the neural responses across the population. So the NDT is essentially processing all the neurons jointly, so it provides like a good kind of comparison point for our separable transformer architecture. So this is, in this case, a supervised version of this, and this is our model, the embedded interaction transformer, EIT, tested in a few different ways, which we don't have too much time to go into. But so in this case, we're interested in basically, so we have electrode arrays that are implanted in motor cortex and also premotor areas within 2 different non-human primates. And it's a relatively simple task, a reach decoding task, where they're making center outreaches to 8 different targets. So the goal in this case is to decode which of the different directions the animal is making a reach towards. This, of course, is an important sort of setup and application, like in brain-machine interfaces, where you'd want to be able to control a cursor or an arm or prosthetic using, brain activity alone. And so in this case, we're asking how well can we translate neural activity during this reaching task into 1 of 8 different target directions. Here I'm just looking at over longer sequences. So if we, for our temporal transformer, if we feed in a sequence of 6 as just like the full context that it sees, versus actually a pretty short sequence. I mean, you're streaming through the whole data. So you eventually will see and train over the whole sequence, but this is just kind of having a very short context window over which to learn from. Okay, cool. So I guess, we compared against these different models. We find that in general, in training on a given data set, we have comparable performance with the NDT, right? So something that can use all the population activity, model it jointly versus the separable, and in some cases we do outperform the NDT through this case. Okay, in addition to this classification task, we also studied a regression task. So this is also movement from motor cortex. And here we're considering a velocity and position decoding. And this is data from the neural latence benchmark, which was a benchmark effort that Chetan and I put together, his lab led. And so this is a number of new data sets that we're really excited to provide for the community to try to kind of standardize a lot of benchmarking efforts within the field. So here we're just reporting the R squared, so the decoding performance. And again, we find comparable in some cases to the NDT, which is state of the art on this particular benchmark. And then, in some cases, we can outperform the model. But overall, the goal isn't just to build a better neural decoder, but it's really to kind of tackle some of those earlier challenges that I talked about before, which is the ability to actually transfer the model. And so now what I'm showing here is the case where we train on one brain. And then remember, we learned the first part, the F and the G, and now we can just use the F to get embeddings of each neuron. And then we can basically just train a linear readout from the new set of neurons that we feed through the model. So we trained on 100 neurons, we could pass in 2, right? It doesn't necessarily matter. We're gonna pass those through, we're gonna concatenate them, and then train a linear readout from that. And so basically what we find is that, okay, I guess, sorry, let me, okay, so here, this is just denoting which brain or which data set we trained on. So here, we're training on one animal, Chewy, and then here, across the columns, we're testing on all of these other conditions. So here, this is the case where we train on Chewy, and then we're actually testing on a different animal entirely. What's exciting about this is in some cases, this pre-training and transfer onto a different animal or onto a different session can actually do better than the model that's trained directly on data from that session. So it sort of highlights this ability of maybe through pre-training over large data sets, we might have the ability to transfer with very few amounts of labeled data and really enable huge, these large pre-trained sort of foundation models, if you will. Okay, so this is a step in that direction. This is an exciting advance because it also allows us to to transfer and to also potentially, resolve more information about what individual neurons are doing as well. So I don't have, so we need to end soon, right? It's at 9:15-ish? Okay. I haven't gotten through the first half, so it's, it's good to go, to discuss things, and it's good to hear the questions as well. Okay, so I'm not gonna, we can also apply this in sort of generalization tasks where we have targets that we've never seen before, and you can actually transfer in that case. But in what remains for the last few minutes, I'm gonna talk about, or the hope was that I would talk about another story, but it's kind of related to this, and so I'll lead into it, and we can discuss more offline if you all are interested. So as I said, in addition to having this ability to potentially transfer, we also have this really nice property where now each neuron kind of has its own distillation of what it's like, and what it does, or how it responds to different inputs. And so we have started to also think about this as a way to look into, what are the individuals doing in addition to what is the population doing in this case. And these are just some pictures of, kind of looking at whether or not neurons that have similar tuning profiles actually do indeed have similar embeddings within that first part of the neural network. And we do indeed find that we have some ways of characterizing similarity in terms of these embeddings. Okay, so then, along those lines, we also have a growing interest in understanding what individual, or actually the function of different cell types, which we know, molecularly have really distinct signatures. So not all neurons are created equal, right? The picture that I was showing initially, where we just lump all neurons into, a population and just, form some sort of representations of them, it doesn't respect the fact that there are different cell types, or maybe these cells are interacting in different and unique ways. And so what I have just shown gives us a way to maybe start thinking about, okay, how can we see different neurons and maybe resolve their cell type, or resolve other functional similarities, other functional properties that they may exhibit. So in another project that's recently just out in Cell Reports, we have also been tackling this question of the function of underlying molecular cell types. And we also use attention. It's not a transformer, but attention is all we need, maybe. And the idea behind this model is to behind this project really is to be able to understand whether or not we can resolve these different molecular cell types based purely upon the spike timing and just activity, the in vivo activity from neurons and intact circuits. And this has been an outstanding challenge. People have done it within in vitro preparations, or where you can actually inject currents into cells in kind of artificial ways. And if you inject certain stimuli, you can resolve cell types. But it was actually still not or it hadn't been resolved to the level that we've done in this paper, like how much you could do this from in vivo activity. And so what we do is we take a really a long time course over which we've measured neural activity. We've done this now, so this is all in visual cortex from the Allen Institute, but the idea I think could be applied more broadly. So both in cases where you have like different visual inputs coming in, so you have trials, or you could take a long sequence and kind of block it up into bins, kind of as we saw before. So what we'll do is we'll take a chunk of time and then compute the distribution of the interspike intervals. So essentially asking, what is the probability of seeing two spikes within a certain distance of one another? So that's sort of shown here. So you have your ISI, and then from that, we're basically going to try to figure out whether or not there are both local fingerprints, so things in the timing information locally that is indicative of a given cell type, as well as whether or not there are kind of global patterns that exist across many of these little instances in time. And what we do is we built basically a way of decoding cell type using both like extracting these local features and then using attention to basically decide which trials or which points in time are really important for a cell type's identity. So it allows us to like get rid of a lot of the stuff that isn't indicative and kind of zoom in on the things that might matter. We applied this in 3 different sources of data. So a biophysical model of V1 where we have like a lot of data and 16 different cell types, as well as calcium imaging data from the Brain Observatory and neuropixels, all from the Allen Institute, so open data sets. So we initially, compared against a number of baselines. I just want to hop to this, which is just kind of visualizing the attention map. So in addition, so you have one computation of your, sorry, what I want to say is instead of just computing one map of which trials or what to attend to, we use a common thing, just a multi-head attention. So you're not just learning one attention map, but in this case you've learned 4. And what we find consistently is that there are attention heads that care about kind of sparse or very specific points in time that are highlighted only over very specific trials. So the bright red just meaning that it's a higher attention weight, and the white being a very low attention weight. So in addition to these heads that care a lot about like very specific trials and points where the neuron is being driven in a particular way, we also learn these more global distribution heads. So something where not any one point in time matters, but it's kind of using a lot of the global context to be able to make its decision. So we've kind of quantified and tried to interpret a bit of this in the paper. But of course, it's still somewhat of a black box, so there's a lot to still learn. In addition, we also tried to interpret what was going on within the model by essentially finding those trials that are most salient or most important for a given cell type classification. And so this is just sort of showing a picture here of finding these kind of relevant firing patterns that are indicative of these cell types. And here we can kind of just use not just average, but a kind of more fancy way of averaging them to find these fingerprints of firing patterns that different cells are sort of maximally, or fingerprints of the timing that seem to be indicative of a given class and maybe not others, or helpful in its determination. So, and so we're really interested in digging into some of this further, because with the 3 different inhibitory neuron classes that we studied, the SST, PV, and VIP cells, they have some kind of unique temporal signatures, and we're interested in digging into that further with our collaborators. So in addition, I think in the end, what's really cool about this model is it can kind of train very flexibly across a lot of different types of stimuli. And so in the end, we actually built a model that was trained both on drifting gratings as well as naturalistic movies, so very diverse presentation of data. And then in the end, we find that combining all of this diverse context is really has this huge enhancement in our ability to decode the cell type. And so this is, I guess, would be state of the art, or we have the ability to do this across so many different modalities. And I think that this also is kind of exciting in terms of our ability to decode cell type moving forward. Okay. And here I'm just comparing those fingerprints across the natural movie and drifting gratings conditions. So we actually find that there's quite a bit of stability in the underlying cell type specific fingerprints. With the drifting gratings being a bit simpler, so we kind of need fewer of these different timing patterns. Okay, cool. So great, that gets us to our summary. So this is grayed out here, but in the bulk of the talk, I discussed the challenges of decoding from the brain and ways that we've been thinking about addressing this. I talked about the embedded interaction transformer as a way to potentially transfer and build large-scale models. And as I said, with this model, we can transfer between animals and sessions, and we think this could be the type of advance needed to, start to build foundational models for neural decoding and neuroscience, which we're really excited about. And then, in a very quick tour, I also talked about a new method for being able to decode molecular cell type from timing and firing information, and how we can do this in kind of diverse conditions as well. And our future work is to, so in the first part, I was talking about populations and then decomposing them, but kind of looking at everything jointly when trying to understand some of these individual properties. And in the second part, I was only talking about individual neurons. So we're really excited to kind of put a number of these pieces together. So, how can you understand cell types in at a population level perspective, and how do you actually use these tools to go further in this understanding of the function of cell types? All right, so that leads me to the important acknowledgments of all the folks that made this work happen. So the first part was work, the EIT was work led by Ran Liu in my lab. The second part on LolCat was joint work with Mehdi Azabu in my lab and Aidan Schneider and Keith Hengen's lab. So we've been collaborating on a number of projects, and really excited that we could kind of, work on these large-scale data sets together. So, and we also have been working closely with folks at DeepMind, and of course our funding and support. And thank you all for making it here early in the morning and showing up. So thanks so much. I guess we have a little time for questions. Yes. Levi.
Great talk, Eva. I have a question about the nature of the recordings themselves. You're talking a lot about identifying individual neural types from each recording, but are they recording individual neurons, or I'm imagining an electrode is in contact with multiple neurons, multiple types of neurons, and maybe it's all convolved together in the actual signal you're working with?
Yeah. Good question. So, as you're very aware of, I at the beginning, we were talking a little bit about spike sorting and how you're not necessarily able to see, exactly or know precisely that what you believe to be a single neuron is indeed. But for the cell type analysis, that's done both with calcium imaging. So in that case, you have different Cree lines where you can uniquely identify the different neurons and their types. And then in addition to that, they use optotagging approaches for their neuropixels probes. So there, as I understand, you'll still have a Cree line, you still have specific cell types, and then you'll use optogenetic stimulation to evoke a potential, and then you can go back and you can kind of associate a given cell type with even the electrophysiological data, but you still have the problem of spike sorting there. So there are a lot of challenges in resolving this. For the Allen Institute data set, the optotagged, there's a lot of quality control that goes into that. But even with that being said, there's a lot of challenges. This is a supervised model, but we're really interested in how to do this in an unsupervised way. So you could imagine, without having to have all that same level of tagging or of all the cell types, maybe it would be a way forward.
Okay, great. Thank you.
Yeah, thanks. Other questions? Cool.
Thank you.
Nice. Thanks so much.